we will start with microscopy. And you might say you didn't sign up for that. But we always teach the class this way because we need it that you are to some extent on the same level in understanding how these images are generated, which you will learn how to analyze. And if you don't have the basics of optics and microscopy, then it will be very easy to stray away from, during your analysis, from the biological truth, or chemical or mechanical or material sciences truth, because you don't know what you're looking at and you don't know what you can expect from a device that generates images. Because of that, we have the first morning always dedicated to microscopy, and I will talk about that. I myself have been doing microscopy for the last several decades, and I'm usually the one who works with colleagues with whom our work group collaborates and advises them on collecting images and carrying out their microscopy experiments the best possible way. And what we will go through is basically these subjects. You have the slides so you know what is going to be the material. So we will go through the principles of building a microscope. Some of these subjects, for example, bit depth and pixel size, etc., Rumen will go through in more details in the afternoon. I will touch upon them as far as they are concerned with the image formation and the analog part. We will also talk about microscopy methods as well as sample types and how to handle them. And then finally, we will talk about how to mark those objects in your sample that you are interested in, that is, how to generate contrast. Okay, let's go through briefly how we use the basic physics of light spreading or the laws of movement, namely refraction and reflection, to generate optical elements, for example, optical lenses. When light spreads as a beam and it arrives at the border of two media, which are optically different, for example, air and water, or air and glass, or glass and water, it will change its direction. This is called refraction, and it basically means that when the light beam comes in at a certain angle, and it arrives from, for example, air to water, like you do on a glass of water or on the beach, it will bend at an angle that is smaller than the angle at which it came. The ratio of these two angles is not by chance, but rather the sine function of these two angles form a ratio which is constant. By constant, I mean characteristic of the two media that meet at this border. For example, if it's air and this is water, then this ratio is 1.33. If this is air or vacuum and this is glass, then this ratio is 1.5. This is called the refractive index. And when I say refractive index and I don't add relative refractive index, I mean the refractive index of the medium relative to air or vacuum, which is considered 1. If I say relative refractive index, I mean between, for example, water and glass, which would be 1.33 divided by 1.5. So it is, in a way, a relative measure. And because this is constant, we can measure the law and prove it, and this was done years ago, of course. You can take, for example, a half circle shape of plexiglass, and then you can illuminate it with a laser pointer, and you will see that the incoming beam will bend towards what we call the optical axis, this normal vector here, this perpendicular line. There will be also some reflection from the surface. So when light comes from a weaker or lower refractive index medium to a darker one, there will be also some reflection. We talk about this later, but right now this is the main point of the message. And this, of course, also means that if I have a piece of, let's say, glass in the light path with parallel lines, then the light beam will refract towards the optical axis, and then when it exits the medium, since the ratio is constant, when I go from an optically denser medium to an optically less dense medium, for example, glass to air or water to air, the light beam will bend away from the optical axis. By the same extent, because the law says the sinus function ratio is constant. So if these two surfaces are parallel, all you have is you shift this beam to there, but they are going to be still parallel. So what a parallel piece of glass will do is simply shift the light path, but will not bend it. But inside the glass, there is, of course, reflection. Now, there's another very interesting, optically very interesting consequence, namely that because the ratio is the ratio between two sine functions, the sine functions max out at 1. This also means that there has to be an angle which, if we reach and we go from an optically denser medium to an optically less dense medium, for example, water to air, there has to be an angle where the light will bend at 90 degrees. Again, we are going from denser to less dense. Water to air, glass to water, glass to air. So there is an angle, a critical angle, above which the light cannot exit the medium. This is called total internal reflection, for the obvious reasons. And this can be calculated simply using the relative refractive index. And this will give you the ratio of 1, and this will give you the critical angle. You can prove it by taking, again, our previous half circle plexiglass or half disc plexiglass. And this means that if you are in an optical denser medium, for example, snorkeling, and you look up towards the sky, you will see your own reflection on the inner side of the water. Because water, air, we are going from denser to less. And some of the beams that reflect from your body or from the body of the turtle will be above the critical angle, so you will get a mirror image of light. But even more interesting, well, more importantly, this is the basis for making optical fibers. Namely, if I have a piece of glass in its most primitive form, and I illuminate the piece of glass with a random array of light beams, there will be many of them that will enter the glass and hit the wall above the critical angle. It cannot exit, so it will reflect. And as long as the fiber was parallel, this will reflect at the same angle, and so on and so on. So I illuminate an optical fiber at one end, and the light comes out at the other end, without exiting in the meantime from the fiber. This is, of course, nowadays very, very often used for internet, so network connections, for example. There are many, many different formations which make the efficiency of such cable much higher than it would be with a simple piece of glass. But this is technical detail. I'll go into that, but the point is that you understand that the simple law of diffraction will be the basis, or is the basis of generating optical cables. And with an optical cable, the light spreads at light speed, as it would. And because of that, the communication is very, very fast. There's another consequence of the diffraction, namely that if the surface is not flat, that what we assumed until now, but bent, then the light starts to behave more interestingly. When you look through a glass of water, you have three optical different media: air, glass, and water. refractive index is 1, 1.5, and 1.3. There's a lot of diffractions going on. So what you will see through the glass is a distorted image of what is behind the glass. Now, this is an interesting observation by itself, but what's more interesting is that I can use this fact, it is that bent surfaces or curved surfaces bend the light in a certain way to generate what we call optical lenses. This is a piece of glass surrounded by most typically a spherical surface on both sides, either convex, sticking out, or concave, which will be sticking in. You will see that in a second. Because what this will do is, if I have two equal radii spherical surfaces, what it will do is it will take a parallel light beam and merge it into one point, a process called focusing. So from this simple law that we just saw, we were able to imagine and then generate an optical element that will focus the light. Now we are getting towards optically useful phenomena. There are as many shapes of lenses as you can imagine. Different radii, different functions that describe this surface. It doesn't have to be a sphere, it can also be an ellipsoid. It can be sticking out, as we said here, so convex. It can also be concave. It can be flat and concave, and so on and so forth. But the main point is that because of the focusing character of such an element, we can use these optical lenses for example, magnifying. We will see in a second why. But the most primitive application of such an optical event is the loop that you might use if you're a stamp collector or if you're an insect collector, which we don't do this anymore, but or just magnifying. If you see this in grocery shops, if you are an older person, they will have a loop attached to the shopping cart, and you can have product underneath it and read the small print. Why is that? It is because these lenses behave in a certain way when they form or allow you to form an image. Namely, if you have a double convex lens, the one that we just quickly talked about, and you imagine that you set it up in such a way that it's perpendicular to the horizontal line, and you place an object into the light path, then there are certain laws with which the image will be formed. So it's not, you don't have to calculate the diffraction angles every time. You can do an approximate rule of thumb type of drawing of how the images will be formed. And this is good enough for now. Basically, what happens is the following. Like I said, when you have such a lens and you illuminate it with a parallel light of B, it will concentrate this beam into one point. Ideally. This is called the focal point or focus point, marked with F. And if the two surfaces have the same radii, then the two focus points are the same distance from the middle of the lens. So it's a symmetrical arrangement. And what the lens will do is the following. If I put an object here perpendicular to this optical axis, then the light beam and we illuminate it from this side, then the light beam that goes through this object and then follows through the center of the lens will not change its direction. This is an approximation. This is called an ideal lens. It has zero thickness. But it's good enough to get an approximation of how the image will be formed. So for an ideal lens, which is, like I said, has a curvature but doesn't have a thickness, this light will go straight through. The light beam, on the other hand, that's parallel to the optical axis, we already know what happens to that. It will be focused. It will be bent through the focal point. So if a light beam comes parallel, goes through the thin lens, it will go through the focal point. So what happens now? We have now two beams, and if I look through this lens from this side, and I follow the continuation of these two lines, they will meet at this point. The extension of the image. So my eye will see what? My eye will see a magnified image of this object. Okay? But a camera won't see this because this image is on the same side of the lens where the object is. So this is a so-called virtual image. I need an observing optics here to see it. But the image is not formed here, the image is formed in your eye, in your brain, in this kind of lens here, not here. Because these beams actually spread this way, there is no light here. So this is called a virtual image. If the image was formed on the other side of the lens, where I can actually put, let's say, a screen or a camera, that's called a real image. But either way, with a simple lens like that, I can magnify things. And this is a loop. This is how a loop works, where you look through it and see the stem or the feet of your insects magnified. The same is true for a lens which has a concave surface, that is, it's bending in. The light that goes through the center of the lens will not change directions, and the light that was going through the focal point will be made into a parallel. So if you look at this drawing, then this you will then observe the light beam that goes to the center doesn't change directions. The one that seems to be coming from the focal plane will be then turned into a parallel beam. And this we can use to generate or draw the formation of an image by any type of lens or any set of lenses. Now I put this here for you for your viewing pleasure. This is just a few examples of how the image formation is depending on what type of lens you use, and also more importantly, where the object is. And there is one I would like to point out, namely if I use the same lens which we used before, and in the previous example we were within 1 and 2 focal points and we saw a virtual image. If I move my object, for example the stem or the fly, outside the second focal point, what I will get is a real image. Now the image will be formed on the other side of the lens. That is, I can record this with a screen or a camera. And so on and so forth. You can look through this if you like, but the most important thing on this drawing is this. Again, where the image is formed and how big it is not random. It's determined by this equation. The equation says that if I know the focal distance of the lens, that is how far is the parallel beam merged from the lens, and I know how far the object is from the lens, I can calculate how far the image is from the lens. Because the inverted values add up according to this equation. Again, this is for ideal lenses, which have perfectly spherical surfaces, but they are infinitely thin. So they don't exist, but it's a good approximation to start your calculation. So if I ask you in the homework, please calculate the magnification of a lens that has a certain focal point and a certain object distance, then you will know this ratio or this equation, and from this one you can calculate the distance of the object and the distance of the image, but from geometry it's a triangle. So the ratio of the distances of the object and the image is the same as the ratio of the sizes of the image. So the magnification is nothing else but the ratio between the image distance and the object distance. Because, yeah, this is a triangle. And this is the magnification. So when you see on a microscope lens 63x magnification, you will know that the image distance from the lens is 63 times as long as the distance of the object from the lens. And those of you who use microscopes, you will know that the object is very close to the objective lens when we focus a microscope. But this means that with a 60px or 100x or whatever, the image will be formed that many times further from the lens than the object is, somewhere inside the microscope. And if we magnify it further with the second lens, then the image will be moved even further away. And this is where your detector is, for example, a camera on your microscope. And from here is just one more step to say, why not put two lenses next to each other? Let the first one form an image of the object. This will form, when you are between 1 and 2 focal points, an inverted, magnified, and real image. And I use a second lens to observe this. Or I put a second lens in front of a camera. And if I continue the drawing, I will see that now I have a second image, which is much bigger than the first one. It is now virtual, inverted, and magnified. So what did I do? If I now have a camera or my eye here, and this is the eyepiece of the microscope, and this is the objective lens of the microscope, what I have here is the simplest form of a microscope. I just built a microscope with two very simple pieces of glass. And this is how it was done when it was first discovered that you can use curved glasses to magnify little creatures like bacteria and single cell living things to generate such a device. Now, we have built a microscope from ideal lenses. Infinitely thin, but surrounded by a perfectly spherical surface. These don't exist, obviously. So we have to deal with real lenses which have limited non-zero thickness. And this leads us to a problem that you will always have to deal with when you do image analysis and you are working with microscopy images, namely that images are not going to be perfect, ever. There will be errors in them, there will be aberrations, as we call them, resulted in by either the lenses, which is what we will talk about, or the sample structure itself, for example, light scattering, light absorption, sample movements, live animal imaging, for example, ultraviolet microscopy. We will talk about these during the next couple of hours, but first let's start with what cannot be avoided, no matter how good of a lens you make, because they come from optical laws, and namely the optical lens aberration. We go through this just very briefly because you don't need to, most of these are compensated for in the microscope, but you need to know about them so you can spot them when you see one in your microscope. The first one and most common one is the spherical aberration. This simply means that if you imagine that your lens is now limited, finite thickness, not infinitely thin, then the refraction of a beam, light beam, which comes closer to the optical axis versus those which are farther from the optical axis, they will not be focused at the same point. They cannot be. The angle will be different and the thickness of the glass will be different. And it is the case, the darker blue beams will be focused closer to the lens than the lighter blue ones. So this means that if you have a finite height object, then the upper part of it, which is farther from the optical axis, will be focused closer, and the closer one will be focused farther. What this means in practice is that when you look through a microscope and you see an image like that, where the center is in sharp focus, but the periphery is not, this microscope and this lens has a strong spherical aberration. And this you will see even the most expensive €1 million confocal microscope that when you look closely at your images and you view the image in its full size, you are not zooming in, you will see that the image information, the pixels that Roman will teach you about in the afternoon, are sharpest in the center and less sharp as you go farther out. This is always the case. It can be compensated for by making this lens into a funny shaped one to compensate for the fact that light which goes through closer to the optical axis would have to see a longer path in the lens in order to be focused in the same way. So a real surface, this is a bit exaggerated, but means that on one side, this will be slightly thicker than on the other side. So this can be done, and when you buy a microscope, it will be coming with objective lenses which are spherical aberration compensated up to a certain extent. Another unavoidable problem, and this you will actually see often in your practice if you are in microbiology or other microscope-related sciences, chromatic aberration. It simply means that light beams with different wavelengths, different colors, will be focused in different focal points. Blue lights, which are the shorter wavelengths, will be focused closer. Red lights will be focused farther. Why is this? It is because the N, the refractive index that I told you about 12 minutes ago, which is the ratio of the sine functions, it strongly depends on the material itself, but also strongly depends on the wavelength of the light. Exactly this way, so that the focal distance for short wavelengths is shorter than the focal distance for longer wavelengths. This is unavoidable. So what happens is, let's say you have two color dots that you want, let's say two molecules that you want to image in microscopy. One is red and one is a combination of blue and green, and you just expect them to overlap. With an objective lens which has chromatic aberration, they will be split. They will be split into a rainbow, basically, because the lens does not focus different colors in the same focal point. And that's a problem. That's a big problem because you can solve this practically only for two, maybe three colors. The solution is drawn here. This is the equation that describes a lens pair which will be so-called achromatic, namely for two particular colors, it will have the same focal distance. But this is only for two colors, because it only works for two refractive indices. If you want a third color, you will need a third element of the lens. It will become a triplet, but you cannot really go further. So when you look at your microscope lens, which you paid, I don't know, 10,000–15,000 euros for, it will have, we'll talk about these markings on the objective lens. It will have a marking which will say, okay, if you look it up on the internet, let's say it's compensated for 600 nanometers and 400 nanometers. But anything in between will still have a different focal point. So all of these lenses will still have a chromatic aberration. So when you do a multicolored biological sample and do microscopy on it, you have to be aware that there is a chance that your blue and green and yellow and red may not be in the same focal point. Which means that in your image you will see them separately, but maybe they are actually overlapping and vice versa. So this has to be checked, and we will talk about this in a minute. With samples or test samples which are small objects with multiple colors, guaranteed to overlap, and then you use this to test your microscope for such errors, such aberration. And a couple more which are much less often observed, but I'd like to mention them. if your illumination comes at an angle not parallel to the optical axis, which is a natural occurrence, the focal points will not only horizontally different distance from the lens, but also vertically. So instead of having one focal point, you will have a focal area. And this means that objects which are optimally little spheres or circles will turn into shapes which looks like a comet in the sky. It has a core, and then it has a cone-shaped part like a skirt, except for those which are smack in the middle of the lens. So when you see this, let's just say you see your molecules in the image showing up like that, then you have co-microphage. This is rare in modern microscopy lenses. And finally, astigmatism. Very, very briefly, just as a background, that you know that light is basically an electromagnetic wave. We talked about it as a beam so far, but as an electromagnetic beam, it has a certain polarization angle. And the polarization angle determines the diffractiveness. So this means that if you have a beam which is not polarized, laser beams are, so you will not see this with a laser illuminated microscope. If the beam is not polarized, then the two perpendicular polarizations will form a different focal point. And what this does is that little bright areas and dots that would appear like this in your image will turn into these laser eye-shaped subjects or images. Those of you who wear glasses and have astigmatism in your eyes, and you take your glasses off at night on the streets of Jena or wherever, you will see this. And when you put your glasses back on, you will see that. I mean, not exactly because I move, but the point is that you will see circles. So this means that you have astigmatism, and they compensate for it with your glasses, and your glasses cost more. This is again quite rare in microscopy, expensive microscopy images, but it could happen, and you should be aware of that. And this is what the lenses do. And they don't mean it, but this is just how optics works. And we try to compensate for them. But as an image analyst, you have to be aware of them because you have to compensate for them if the microscope is not compensating. You have to be aware of them. And when you are a microbiologist, when you do your experiments, you have to be also aware that this is a possibility when you analyze your images. And before you draw a conclusion of, okay, these proteins co-localize, if you label them differently, is there a chromatic aberration? How do I check it? And if you are analyzing your own images, which hopefully you will after the class, you have to know how to compensate, how to shift images to make up for the shift caused by the lens itself. Now, what does the sample do to make the life of an image analyst more difficult? Matters or tissue, cells, etc., they will scatter light. And that's nothing to do with the lens. It has everything to do with the structure of the sample that we're looking at. Proteins, lipid particles, they love to scatter. What we mean by scattering is that if a light comes in from a certain direction and it hits, let's say, a lipid particle, the light will not continue straight ahead, but rather it will spread into various directions. Which directions and what the distribution of these scattered light is depends on the size of the object on which you scatter the light. If these objects are very small, a few hundred nanometers, the same range as the wavelength of visible light, then you will, and you illuminate, let's say, a fatty tissue with visible light, The scattering pattern will be like a number 8, basically. So the scattering forward and backward has the same probability. You can see it better here. And this is called the radius scattering. So again, the objects are a few hundred nanometers, a couple of hundred nanometers in the range of the wavelength of visible light. We'll talk about this in a minute. And you will get a scattering pattern like this. So the forward scattering and the backward scattering is equally probable. Perpendicular scattering is not the same problem. If you then increase the size of the object, now we are talking about, let's say, micrometer size, I don't know, lipid droplets. The forward scattering is much more likely than the backward scattering. This is called the Mie scattering, named after physicists. If you increase the size of the scattering particles even more, the forward scattering becomes more and more likely. So from small objects in the nanometer range, I will get the symmetrical scattering. From large objects, I will get the forward scattering. This means that when you illuminate a thick tissue, a lot of your photons, both on the excitation and the emission side, will be scattered. You lose information. And this has to be compensated for by either cleaning the sample, making it less scattering, or doing image analysis by trying to compensate. And there is an interesting consequence of this one, namely that the relatively large water particles inside a cloud, for example, they result in a Mie scattering pattern, a strong forward scattering. Whereas smaller particles in the clear air will result in a more symmetrical scattering. And as a result of that, you will get a color difference. A mostly blue sky from the radius scattering and a mostly gray reflection from the cloud. And I did this little simulation for you to see that if you have a 500 nanometer size object, so it's a small molecule, a small particle, for example, vesicle in your cell, phagosome, whatever. If you look at it from a micrometer away, you already see a much larger spot than reality. If you look at it from 3 micrometers away, you have basically no idea how large the original object was. And when you look from 50 micrometers, which is not very thick for a tissue, you lost the information totally. This I assume to have a radius scattering to be seen. This is because this is easy to put into a computer. There's a closed formula for this. But if you look from 50 micrometers to 50 micrometers tissue, you lost all the information. So light scattering can really kill your microscope experiments in this regard. Now we talked about how some lenses, or most expensive lenses, are compensated for many of these aberrations, but they also contain more information when you look at a lens closely. It has a lot of mysterious symbols. We will talk about all of them during the morning session. But basically, what I wanted to point out right now is these color codes, which will indicate, for example, is the lens corrected for, I don't know, chromatic aberration, and if so, what pair of wavelengths is compensated. It will also contain information about the magnification. You remember this is the ratio between the image distance and the object distance. It will indicate, for example, that the lens is corrected for spherical aberration. So at least you can see it well if you get this one. And it will indicate other parameters which you don't understand yet. for example, the immersion media that can be used with the objective lens, methods that can be used with transmitted light microscopy, the resolution of the lens, and so on. So we will go through these and learn how these lenses can be characterized in terms of optical behavior. Now, inside such a lens, you don't find a simple structure like a loop anymore. It is a very complex structure where they have, for example, color compensation. You recognize here the color compensation, chromatic aberration compensation duplet. In this case, it's a triplet. You will see the slightly asymmetrical shape which compensates for spherical aberrations. So this is a very, very carefully designed engineering marvel, which will then cost you. So if you go to Zeiss and you want to buy a 63x immersion lens with microscope, this comes in 10,000, 15,000 euros per lens, which also means you want to take good care of it once you purchase them. But inside of this, you will see a complex structure. And of course, various manufacturers make many of them, designed for various purposes, various thicknesses of tissue, colors, working distance, that is how far you are from the object, and so on and so forth. And all the information you can always find on the manufacturer's website. So during the homework, you will probably have to do this to look up lens properties based on when I tell you, okay, size 63x, plan up or whatever. You look it up, and from that you figure out a lot of properties of the objective. And you will need to know this. Why you need to know this? You need to know this because this determines what you actually can do with this lens optically. What can you expect from the microscope that is equipped with one of these lenses? You will see this very soon, that if somebody comes to you that, okay, here is a microscope with a 10x or a 5x magnification lens, I want you to examine phagosomes during phagocytosis in a cell. You said, I cannot do that. This lens is not designed for such high magnification work. It doesn't have the power, it doesn't have what you will learn to be called the resolution. But for that, you need to know how the lenses work, and you know this now, and what these parameters mean and how to look them up. And this leads us to the most important, by far, the most important parameter of an optical system, namely the optical resolution. And what does this mean? This means that the goal of a microscope is to magnify tiny objects so we can see them, because we cannot see them with the naked eye. This also means that the performance of an optical system or a lens itself can be measured by how small is an object that I can still see as an individual object. Or how close can two very small objects be to each other so I still see them as two. Let's say I have a muscle cell with 3 nuclei. Can I see them as 3 nuclei if they are close to each other, or do I just see a big blob in my image? Or I have microtubules, micrometer or so, or I have mitochondria, micronucleation inside. Can I see individual filaments? Can I see individual mitochondria as separate, or do I see one blob? High resolution objective can see them as separate, low resolution objective cannot. But for that, we have to then talk about what resolution is. This is a very, very important part of optics and the lecture itself. The optical resolution will be measured with a parameter called, or will be determined with a parameter called numerical aperture. I'll explain what that is. And then we will talk about how to increase the numerical aperture. And higher numerical aperture means higher resolution, better performing lens. The numerical aperture you will see in your practical biology life will go from, I don't know, 0.05 to a very small one to 1.4 to a very high resolution, expensive lens, a factor of 30 easily. And based on that, we will learn how to calculate the resolution of a microscope and how to improve it with various tricks. So what is numerical aperture, NA? It is a very simple formula, similarly to what we had before. It uses the refractive index, which was what we talked about earlier. And it uses an angle, which is the angle of light that enters the front of the lens. So if you imagine that this is an objective lens, like the one we saw in the previous slide, cut in half from size. And this is the front of the lens. If you remember, there was a small lens at the very right side, which was it's called the front lens, which is the one that's in contact with the sample itself. And down here, we have like a whole bunch of other lenses. So this is the one that we make contact with. And here there is a metal like brass or aluminum or stainless steel cone within which this lens lives. Then there is a maximum angle at which light will be able to enter the lens. Anything above this angle will hit the coat and then it will not enter the objective. Yeah. So this means that if I measure this angle, plus I know the refractive index of this part here, I can calculate the resolving power of a lens. This means two things. Very important. One is that the resolving power of the lens depends directly on the refractive index. So it doesn't matter what we have here. If we have air between the lens and the sample, it's the microscope cover glass. If we have air here, that has the refractive index of light. But if I put, for example, oil here, oil has the refractive index of glass, 1.5. If I put oil here, just a drop of oil, I will increase my numerical aperture by a factor of 50% right away, just by putting a piece of oil. You will see in a second part. Or if I put water here, that has an N of 1.3, I increase my resolution by 33%. And the other factor is this. So the larger this cone, the higher the resolving power of the lens. So why don't we move the sample closer to the objective lens? That's by far the easiest way to increase this angle. So lenses with a shorter focal point, where the sample is closer to the objective, have higher numerical aperture. So this one is a better lens than that. Otherwise, they are not different. Now, how can I then improve the NA further by putting something in between the lens and the object? Yes?
What was the objective again? The lens itself or something else?
So the objective, this here, and this is the front lens. And this is the sample. So you have the sample on a microscope glass. You put your cells in it, cover it with a cover glass, and you put it on the microscope. So this is upside down. Yeah? So if you can focus closer, you increase the angle and you increase the NA. Okay?
I mean, what does the objective stand for? Is it the sample itself?
I'm sorry, I didn't clarify that. My objective from now on, I mean objective lens. That is the lens that it's not one lens, it's an array of lenses like you saw in the slide that is used by the microscope to form the image. Sorry. So I mean objective lens. Yes. So what can I do about this? Well, what happens is that you know this already, that when you come from the biological sample to the glass, thin glass cover here, and then you let the light come out, then it's going from a higher optical density to a lower optical density, higher refractive index to lower refractive index. That means that the diffraction is away from the optical axis. Right? It's refracting away. So first of all, eventually you will have total internal refraction, so not all the light will exit the sample. But second of all, the light that comes from the sample, a lot of the beam will go at such a high angle that they will never reach the lens, the front of the lens. So I would like to collect these light beams and direct them into the objective lens. How do I do that? I put something optically denser in between the sample and the front of the lens. Why does that work? It works because, first of all, on this surface, between the cover slip and this medium, it's called the immersion medium because the lens immerses into it. The refraction will be mostly eliminated because now I go from a refractive index of 1.5, which is the glass, to oil, which is refractive index of 1.5. So the light beams that like to refract away will now happily stay inside this droplet of oil. What did I do? I increased my NA because I increased my refractive. I can also use water. It depends on the lens, how the lens is built. Water works well, but not as well because now we have a transition between 155, which is the glass, to 133. So I lose more light here than I lose here, but I lose less light here than I lose here. So now you know that the numerical apertures, if we have a contest, this will be the winner. Second place, third place. And sure enough, that is the conclusion. So now if somebody says, okay, I would really like to see my individual mitochondria, I have a choice between a 63 or 40x, what we call air objective, where there is nothing in between samples, a 40x oil and a 40x water. And I'm looking at small objects, submicron objects. Now you know which one to choose. You will not go, oh, I want the air one because then I don't need to use oil. Oil is messy. Oil is messy, yes, and you have to clean the objective lens carefully in between experiments. But you gain a factor of 50% in terms of resolution. Okay. Now, if microscope only has a water immersion lens or a glycerol immersion lens, they also exist. That's also still better than the air. The air objective, of course, has the advantage that you don't need an immersion medium. So it's faster. When you change your samples, it goes faster. You don't have to clean the sample, you don't have to clean the objective lens every time, put oil on it, make sure there's no bubble in the oil, and so on and so forth. But there is a trade-off. You get better resolution. And now we know why. How can then we precisely calculate this resolution? Well, we need to know the numerical aperture, and now we know how to calculate that. We need to know the wavelength of the light that we are using. We'll talk about this in a second why. And from this one, we can simply calculate the power of resolution of a microscope using this formula. This called the Rayleigh criteria. It basically means that the resolution is directly proportional to the wavelength of the light. What does this mean? It means that blue light, which has shorter wavelength, has the potential for a higher resolution, a better resolution than red light. And that's very important when you design your experiment, for example. If you say, I want to count nuclei, and I want to see individual microtubules or mitochondria or very small objects. What color should I use to label? We talk about fluorescence labeling, but you already probably know what that is. What color light should I use to image them? And now you say, well, the wavelength for blue light is much shorter, so it has a better resolution because of this formula. So I want to have my small objects to be blue and my large objects to be red, if I can. Unfortunately, of course, the most typical nuclear DNA label is blue, which is the most unfortunate color for such a large object. If you want to resolve DNA RNA clusters, of course, then it's advantageous to have blue color, therapy or Hucht for your label. But if you are just counting them and you have an orange or red label for nuclei, by all means use it. You will not suffer because they are micrometer large. And then use the lower wavelengths, the blue ends, to the smaller. And the other thing you should do is increase your numerical aperture. You are dividing by it. Now, in the formula, you will have two values, the objective lens numerical aperture and the condenser lens. I will show you what condenser lens is, but basically, remember we built a microscope not just from one lens, but two lenses, the second lens observing the image formed by the first lens. The first lens that's directly in contact with the object is the objective lens, and the second lens is the condenser lens. And this is a sum of the two. This has a very important consequence for practical microscopy. That optimally, this value and that value should be both very, very high. Practically, this is not going to happen because condenser lenses are usually of lower quality, lower magnification, and much lower NA than the objective. So for really high resolution microscopy, when we talk about super resolution, this might come up. You may want to have a microscope where your second lens is just as good as the first one. Problem is, these lenses cost you 15,000, these lenses cost you 800. So if you don't have the budget, you cannot have both of them. So basically what will happen is that the resolution will be determined by how good your objective lens is, and will not depend so much on the quality of the condenser. But this means that, at least as a rule of thumb, we already know that our resolution limit will be around 200, 250 nanometers. This is assuming that we are working with visible light, which goes from about 400 nanometers to 700 nanometers. And we use objective lenses between 0.8 and 1.5 numerical, 1.4 numerical. But the concrete value will depend on a lot of things, as we will see later, but as a rule of thumb, again, 200, 300 nanometers is your data, if you are using visible light. This is already extremely important because if you say, I want to see, I don't know, nanoparticles that are 100 nanometers, and I use nanoparticles to deliver drugs to a tissue, cure the liver, whatever. You cannot see them. You will not be able to see individual nanoparticles because they are too small. They are below the resolution of the microscope. On the other hand, if you say, again, my example, I want to just count nuclei. They are, I don't know, 2 micrometers diameter. Then you don't need the best microscope and the best lens because you need a resolution of, I don't know, 500, 600, 700 nanometers to see them. So you adjust your experimental conditions and your microscope selection based on what you want to observe. You don't always need, for example, super resolution microscope, which we will talk about later, where the resolution is even higher. Sometimes, but you have to be aware of what you actually need. Prices, how much you pay for a system, how often will you use it, what are going to be the typical usages, will determine what you actually need. Just like we don't drive a Lamborghini to go to the clinic, to go to work. We drive a Volkswagen, whatever. You don't need, what am I going to, I would love to have it, but what am I going to do with it in those cases? Okay, why is there such a thing as a limited resolution? The reason is called light diffraction. This means that the way light behaves and how we learned about it until now was that if there is a parallel light beam, it just goes on forever and it carries the information with it. If it meets a lens, it will be focused or diverged, whatever the lens does. But otherwise, the information that the light carries stays with it because it just spreads through the optical material. But this behavior changes dramatically when the light beam arrives at an obstacle or it arrives at a very small, it shines through, I should say, through a very small object or a very small gap, for example, in a piece of metal. If it meets an obstacle, which is approximately the size of the wavelength of the light. Above that, when the light spreads through microscopic obstacles and molecules and structures, it just spreads as it has before. But when it arrives at a very small area, or it's emitted by a very small object, which is the size, like I said, approximately the wavelength of the light, then a very interesting phenomenon occurs. Namely, the light beam that was spreading happily until now arrives at a gap, for example, in a metal film, and the size of this is measurably close to the wavelength of the light. Then this small area starts to behave as a light source itself. So when the light arrives at this point, it doesn't know anymore where it came from. It starts to spread spherically in every possible direction. It diffracts. So a good experiment is if you take a laser, for example, and you illuminate a screen, and then you shine the light through a hole, you just see this spot on the screen. But if you make the hole so small that it's just less than a micrometer, then on the other side of the metal, you will see a pattern. You will not see a spot anymore. And the reason for that is that a parallel beam will behave, so this spot will behave as a source of a lot of individual beams now. So the light that carried the information up to this point starts to spread randomly in every direction. So what this means is that when you get through this object, you forget all the information that you have been carrying. Where you came from, what was if you are an electromagnetic wave, what was your phase? This is all gone, and instead you have an array of random spherical waves spreading on the other side of the obstacle. So this means that as long as we are using visible light and we are examining objects which are sub-micrometer, this is my warning that in 10 minutes I should give you a break. everything is fine. But once I try to use the light to carry information about objects less than their wavelength, I have a problem, the problem of the diffraction. And this means that if I have a very small object that emits light, and by small I mean approximately or less than the wavelength of the light that we are using in the experiment, then, and I form an image with an objective lens. And here I have, let's say, I don't know, fungus, micrometers. And my objective resolution is 2 micrometers. It's a cheap lens. Then what I see on the other side as an image is not the image of the small object. Instead, what I will see here is a pattern like this. What happened here was that this small object, this small molecule, for example, ejected or emitted light, which went through diffraction, and as a result formed an interference pattern. So there is a small spherical object here, and here there is a pattern which has nothing to do anymore with what I imaged. So when I look at this pattern and I ask you, okay, what was I imaging? How big was it? What shape was it? You have no idea. If the size of this object goes higher and reach the resolution limit, then you will have a proper image. But with smaller ones, you will have a pattern. This pattern is called the airy pattern, and the first maximum here is called the airiness. So this means that when you have, I don't know, an extracellular vesicle, 100 nanometers in diameter, and you put it in a microscope, and you realize that 100 nanometers is below the resolution of the microscope, then what you expect is that from each vesicle, you will not get a point-like image that would actually describe the proper size and shape of the vesicle. What you will get is a diffraction pattern. So I cannot image such a thing anymore because I don't know how big it is, when it is. I just know that the center of it is somewhere where the center of the pattern is. So I can still localize it, even if it's very small. But I cannot tell you how big or what shape it is. This will be very important when we talk about single molecule localization microscopy in super resolution active planes. That I can localize it, but I cannot see them. Now, this means that a good lens, a higher NA lens, will have an airy pattern which is relatively small. A worse lens, which will be the relatively low numerical aperture, will have an airy pattern which is relatively large, because the resolution of that lens is worse. So if I look at this pattern, I will say, okay, this so-called image is nothing else but what the microscope formed when it was trying to form an image of an object which is smaller than the resolution of the microscope. I got an interference pattern, not the actual image. Now, if I increase the numerical aperture, then the size of this pattern and the center of it, the airiness, will get smaller and smaller. NA 0.2, 0.8, 1.3. Now, if I have two objects which are close to each other and they are both below the resolution limit, how far they have to be so I still see them as two? I know they have to be farther than the resolution of the microscope, but in terms of the airy pattern and the airiness, how can I describe that? Well, imagine the following. You have one object below resolution, what you see in your microscope is this interference pattern, the area. So I know that my object is here, but I don't know how big it is. If I have two objects which are very close to each other, they will both form an area pattern, but what I will see in the microscope is this, a blob. And I will not know how many components there are in here. However, if I separate these two small objects far enough so that the area patterns, the first part, the center bright part of the area patterns are separate, then I can say this is 2. Because there is one pattern here, one pattern there, and there is a gap between. I can say that these are 2 small objects, both below resolution, but there is 2 of them. I can count them. So I resolve them. Okay. So in this case, I cannot resolve the sample. In this case, I can. It's not optimal. There is no full separation, but from comparing the area patterns, I can say that I can count, in this case, how many molecules there are. I have no idea here. Okay? So when I go up with numerical aperture through this way, eventually I can get to a point where I can see individual objects. What was the condition? The condition was to have this area pattern small enough, narrow enough, thin enough, so that these 2 objects separate. Now, this is fine. This tells us how close these objects can be so that we see them separately. But then the question is, what resolution should the image itself have? That is, how detailed the image should be that I take of this object. Rumen will talk to you about pixels and pixel sizes, but for now it's enough to say that when you generate an image and when you look at it on your computer screen, you will look at it on a digital format where the image is made of image points, so-called pixels. Now, let's say your microscope is good enough so you can resolve 200 nanometers in terms of spatial resolution. If you then have collected your images in such a low resolution that each image point, each pixel is 500 nanometers, can you resolve this 200 nanometer object? You cannot, because the pixel itself is bigger than the resolution of the microscope. So if I tell you, I have an image where the microscope was set so that the resolution is 200 nanometers, what resolution should the image itself be? Then you will know it has to be small enough, the pixel size has to be small enough so I can actually see these 200 nanometer resolved objects. And a very simple rule of thumb is that if your resolution is a certain distance, let's say 200 nanometers, then the pixel size should be half of that. So in this case, 100 nanometer pixel size. If your microscope is not capable of that, because let's say it's a camera-based one and you simply don't have the camera chip good enough to have such a small pixels, then you cannot take advantage of the resolution of the microscope. So optically it works, digitally it doesn't. The principle is called the Lundquist criterion, and it basically says what I just mentioned. If you have a certain periodicity in your sample, in this case the resolution itself, then you can only restore the physical properties of the object from a digital image if your sampling rate is at least twice that of the actual sample. This means, like I said, that approximately half the size of the resolution in terms of area. This means that if your pixel is too large, then you will not see the resolved image. If your pixel size is too small, there is such a thing, then of course you will see the details of these objects very, very well. But in order to collect such small pixels, you may need to oversample your data, which means that eventually you will, for example, illuminate the sample too long, and you simply will kill your cells. It could be photo damage, you could be bleaching your photo dye, and so on and so forth. And it might take much longer. So there is an optimal setting for this one. When you determine your microscope settings, how large should my pixels be? And in the afternoon, you will see how this works in practice. So basically, when you have an expensive microscope, it will also, the software will tell you how large the pixel size should be, assuming it's settable. And if not, then you can set it up yourself by using, for example, a zoom option. Now, so far we learned that the microscope will never return an optimal depiction of the object. There are optical aberrations, and there is a limited resolution. There's light scattering. So the microscope will do something to our image which will make the image less than optimal. But what I would like to have is, if I know the resolution of the microscope, and I know the optical properties of my sample, I would like to know what the optimal image would look like, despite all the mistakes that the microscope adds to the image. So I would like to know that if this is my sample, the real object, and this is what I see in my microscope, what did the microscope do to my image, and how can I then, from this image, calculate what the actual object looks like? So for this one, I have to be able to characterize the optical performance of my microscope. And this means, for example, the need to know the shape of the airy pattern that we talked about. What sort of an image form is formed by the microscope when I put a sub-resolution, very small object in the microscope. And this can be described with a simple function called the point spread function, PSF. And it does exactly as the name says. It tells you that if you put a point, a perfectly spherical object, in the microscope or under the microscope, with a size below the resolution, what will the image look like? How does it spread the image of a point into a real image? And if I know this function, and I use a mathematical term called convolution, you will hear about this later in the afternoon, I can calculate what this measured image will look like. But that's not the important part. The important part is that if I know this function and I know the image, I can also calculate the object. So knowing the point spread function and knowing the image, I can calculate back what the object would look like had the microscope not distorted it. This process is called deconvolution, and this is the first step of image analysis, where you either you do the experiment yourself or you ask the experimenters to do it to measure the point spread function. We will talk about this after break. And using software which you can have access to. Right now, JIPipe is being worked on to have this option to calculate back the actual shape and size of the object, taking into account what the microscope does to the object. And a couple of properties before we stop for a few minutes. By knowing the numerical aperture and the optical properties of the optical elements of a microscope, you can always calculate the point spread function. So you can always estimate what the microscope will do, even if you don't know your sample. Of course, this means that everything that has to do with the sample, like light scattering, will not be known, and that's not a perfect solution. So, in practice, what you have to do is you have to measure the constant. You measure it simply by putting a sub-resolution, perfectly spherical object into the microscope and take an image of it. And the image will look like that. And that means that a sub-resolution subject will look like that in the microscope. That is your measured point spread. And if you compare this the theoretical one with the measured one, very often you will see a big size difference because of the properties of the sample that it affects the microscope image formation. And then your deconvolution, your compensating for it, will be much more efficient with the measured one than with the theoretical. The measured point spread function will also depend on how scattery your tissue is, which also means that if you measure deeper in a tissue, for example, this is from a mouse bone marrow, the deeper you are in the tissue, the larger the point spread function. This means that if I focus deeper into a scattered tissue, my resolution will drop. Your information from the part of the tissue that's close to the objective lens will be much better resolved than the information that you get from deeper in the tissue. Eventually, you will lose the details when you focus very deep, for example, in brain tissue, liver. They are very scattered, bone marrow in this case. Eventually, you will lose the resolution. You will also see microscope images if you do an axial collection of images, that the detail, the contrast is just gone when you focus deeper. And this is an example. These are fungal hyphae that grew in a tissue. And this is what the raw images look like. This is just with a regular microscope. And as you see, those which are in focus of the hyphae are very sharp, but those which are not in focus also appear as a blur, and that's the result of the point spread functions. It turns an object like this into a cone shape or a cigar shaped object, which means that the farther you are from the focal point, the wider the image becomes, so you get this blurred image. But then, by using, in this case, even theoretical point spread function, you will get a sharp image. This is the same image as on the left side after deconvolution. So here, the microscopist didn't have to do anything other than collect these images, give it to us, and then we deconvolved it with using theoretical point spread function, and we got an image which is now perfectly analyzable. If you try to analyze this, you will fail because you don't even see the object. So this is the very, very first part of image analysis. This is now the axial view. The first step that we usually then start with. This is bone marrow before and after deconvolution. There are two basic configurations, upright and inverted microscopes. An upright microscope will have the objective lenses above the sample. This is the general structure. This is where the observant's eye go to look through the eyepieces. These are the objective lenses, the sample itself, the condenser lens, and the illumination comes from either side of the sample. As you will see, and you probably have seen microscopes before, for practical reasons, usually there are more than one objective lenses installed on the microscope, so you don't have to take one off to be able to use a second one. They sit on a rotatable turret. The condenser lens, which is positioned on the other side of the sample, is what we talked about, that it's usually a lower quality lens than the objective lens. In certain techniques like two-photon microscopy, oftentimes they put an equally good quality lens down here, And in this case, for example, if you have an oil immersion lens for objective lens, then you also have to have an oil immersion lens for the condenser. In terms of the light source, this can be a simple light bulb for transmitted light images, we talk about this, or LEDs or lasers for confocal microscopy. There are two different pathways for a microscope, either upright or inverted. The reflected path or epifluorescence path, it's basically a clever solution for the problem of having to have a high quality condenser lens to take advantage of the numerical aperture. Namely, we can say, why don't we use the same lens for both illuminating the object and collecting the light from the object? And if we do that, then we get this reflective epifluorescence light path, which means that basically you use a light source, you reflect it through a mirror, we'll talk about this in a minute, through the objective lens onto the sample. You generate, for example, fluorescence, which we will discuss in a minute, but then you collect the light produced by the sample through the same lens. This means that this light then goes up and into the detector, or the eyepiece, or the camera, or whatever you want to use. But this means that in the numerical aperture, the resolution formula, where you remember we had NA of the objective lens plus the NA of the condenser lens, both of those NAs now will be produced by the same lens, so you only need one high quality object. In the other configuration, we talk about inverted microscopes, where the objective lenses sit underneath the sample. This has the advantage, of course, that the top of the sample is accessible. So this means that if you want to change solutions on your samples, you can easily do that. On an upright microscope, you cannot, at least not easily. The disadvantage is that if you use immersion objective lenses, which we talked about, then the oil or the water is put on the top of the objective lens, which is sitting basically upside down. So there is a chance that you lose your immersion liquid because it just flows off the objective lens. Here, I've shown another light path where we illuminate the sample from one side, but we detect it from the other side. It's called the transmitted light path. This we do, for example, for bright field microscopy images, where you simply use unlabeled samples to image, for example, to observe cells migrating in a tissue or in a petri dish. A detector is then positioned at the end of the light path on the other side of the sample. And in this case, the total NA is determined also by the numerical aperture of the condenser lens. So here there's a slight drop in the total resolution of the microscope compared to the epifluorescence or reflected light path. Now, we will mostly talk about the epifluorescence or fluorescence light path and fluorescence microscopy, but very briefly I wanted to talk to you about transmitted light microscopy. Also because I would like to emphasize how much information you can still get from this modality, despite the fact that you do not actually necessarily label anything in your cells. And basically, there are three main fields: bright field, phase contrast, and differential interference contrast microscopy. We will not have time to go into the details, but the main difference is between how you generate contrast. In bright field microscopy, you simply let the light go through your samples, your cells, and then the more absorbent parts of the tissue will be darker, the less absorbent ones will be brighter, and that generates a simple contrast. It can also be colored samples, which are naturally colored, and then use a color camera to capture the image. In phase contrast microscopy, you basically emphasize the edges of objects, so you will see the membrane very well, and also you will see the membrane of the intracellular compartments. And in differential interference contrast microscopy, you generate contrast from very, very thin samples, which could otherwise not be seen even in bright field microscopy. For all of them, it's absolutely important that you align your microscope. As you can imagine, you have a light source, as we saw it before. You have a condenser lens, you have a sample, and then you have the objective lens, and then you have the detector. If these optical elements are not lined up, then your image formation will naturally suffer. There are alignment methods, the most common being the so-called color illumination, where you align, you use apertures which are in the light path to make sure that the alignment is as optimal as possible. For each microscope that you use, there will be printed instructions of how to carry this out. But what I noticed with many biologists or colleagues, I should say, I work with, is that they are not aware of the fact that this has to be done before each experiment, because the illumination path depends strongly on the sample itself. So if you don't check the alignment of the microscope, you will not get your optimal transmitted light microscope images. Again, without details, this has to be done every time you change your samples or you change your objective lens. Now, as a result of that, you can get high quality, high resolution transmitted light images, where you can observe, for example, here you see neutrophils in a petri dish migrating, and you have high enough resolution without any label to be able to identify every immune cell and then to track them. And then you can measure how fast they move, for example, in the presence of pathogens or without pathogens. Do they move towards pathogens? How their speed depends on the activation status of the immune system. These are neutrophils from human blood. Or you can say, how do macrophages, the large cells, and neutrophils, the small ones, in the presence of fungi. And what you will notice, for example, is that the cells, the macrophages, collect the fungi. These are the green labeled objects. But the macrophages themselves are not labeled. This is just a very well aligned microscope in transmitted light without any labeling. And then you can observe important events, for example, macrophage-based phagocytosis, or the death of a neutrophil that you observe here, where the dead neutrophil gives its content, which are fungal spores, to a nearby macrophage. So the fungal spores are not released back into the system, but rather given to other immune cells. And this image itself was basically a free gift from the microscope, because transmitted light microscopy doesn't require any special preparations, except aligning the microscope itself. For phase contrast microscopy, you basically illuminate through, instead of homogeneous illumination, you have an illumination pattern which looks like a donut. So basically, you illuminate through a circle, the center of which is dark, and then in the microscope objective itself, you have a similar pattern with the exact inverse. So where there is a gap in this ring, there is a true blockage in the faceplate, in the objective lens. So this requires a lens which is capable of phase contrast. So on the labels on the side of the lens, there will be a pH label indicating that this is a phase contrast ready objective lens. And the effect of this is this: if you look at a sample, this is a blood smear, with bright field microscopy versus turning the phase contrast on, you will see that the membranes are very well emphasized here. And this can be much more easily counted and characterized than the simple bright field image. Alignment is again, of course, crucial. So the two patterns have to be perfectly aligned, which has to be checked again before each experiment. And finally, about differential interference contrast, we mentioned that the refractive index and thus the optical behavior of the light depends on the polarization angle of the electromagnetic beam. This technique basically splits an unpolarized light into two 45-degree separated polarized beams and takes advantage of the fact that the refractive index is dependent on the polarization. So it means that the two beams, one of which will go through the sample and one of which will not go through the sample, will have a different arrival time to the other side of the sample. So when we merge them together, they will form an interference pattern. And as a result of which, we will see these pseudo 3D relief patterns, which is very much easier to analyze than a regular bright field image. So for example, here, which I just got from the internet, you see the internal structure of this one cell organism without any label. This is just using DIC microscopy. How do you know if your microscope is capable? The lens itself will say on the side labeling that it is a DIC system. And various cells which are otherwise would be, for example, fibroblasts are very, very thin, would be very difficult to see them without generating this interference contrast. In addition to transmitted light microscopy, of course, usually we label the elements, the objects that we want to image. The most common form of that is using fluorescence microscopy. The principle of that is quite simple. We have a microscope, we have an illumination light source, and then we illuminate our sample, and the sample is labeled with a molecule that will emit light depending on the excitation of the light source, the excitation wavelength, and this will give you information about the size and location of the object that we label. Now, why is that possible? It is possible because when you look at the energy structure of a molecule, if you illuminate such a molecule with a photon, the energy of which corresponds to one of these transitions between the basic energy state of the molecule and its first or second or third excitation, this molecule will be able to absorb this photon. So you illuminate the molecule, the light arrives at the molecule, and then it's gone. What happened to it? It got absorbed by the molecule because the molecule had an energy transition that was able to match the energy of the photon. Blue photons carry more energy, of course, that's why you get sunburned. Red photons carry less energy. So depending on the color of the light that you use, you may or may not have photons that are going to be absorbed by a certain molecule. Now, this means that this energy absorption event depends on the illumination and on the molecule itself. So you have to design both of them. And you have to know the characteristics of both of them if you want to design your microscopic experiment. What happens later? So let's say we have a photon which matches one of these energy levels. The molecule gets into an excited energy state, but it doesn't like to be. If you ever studied quantum mechanics, you will know that excited energy levels are not stable. With a certain probability, the molecule will try to go back to its base ground energy level. This probability is quite high, so the transition back to the ground energy level is quite fast. When this happens, we will see the emission of this energy that is released when the molecule relaxes from its excited state back to its ground state in forms of, in many cases, a photon. And depending on the energy structure and the wavelength of the light that we use to excite this molecule, this emitted photon can also be a visible light photon. So we can see it. And if you can see it, we can detect it, we can put it in a microscope, and we can use it to label particles with these molecules. Certain detail, just to make this a bit more realistic. When a molecule is in excited state, these excited states are not just one very, very precise energy level, but rather a range of levels. And the molecule will try to relax itself by transitioning between these sub-energy levels, which means that the energy that is eventually released in form of this fluorescence photon is a little bit less than the energy that came in. Because of this so-called internal conversion, it's a self-relaxation of the molecule. This means that the color of the incoming photon will be different from the color of the outgoing photon. If you had a blue photon coming, I got the colors from here. If you have a blue photon coming in, you will see a green photon coming out, because green has less energy than blue. If you have a green photon coming in, you will have a red photon coming out. So this means that during fluorescence, I will generate an emitted light which is slightly longer in wavelength than the light that came in. Now, if you have a large molecule, then of course these energy levels are even more complicated, which means that there are many, many possible transitions, both for absorption and for the emission. So a lot of various wavelengths, blue range light photons can be absorbed, and you will get a range of green photons, not just one wavelength. This means that these molecules, which are able to generate fluorescence light, are characterized by a certain spectrum for both absorption, excitation, and emission. There is not only one wavelength that will excite a fluorescent molecule, but a whole range of wavelengths. And there is not only one wavelength that comes out as fluorescence photon, but a whole range because of these transitions. So this means that if you take a fluorescence molecule, for example, FITSI, which is very commonly used, or Alexafluor 488, which are optically very similar, and I ask, okay, what wavelength should I use to excite this molecule? Then I will say, well, this is the so-called excitation spectrum. It's basically a curve that describes how likely it is a photon to be absorbed by the FITSI molecule or the Alexa molecule. It says if you use a 400 nanometer violet light, it's not going to get absorbed. If you use 600 nanometer red light, it's not going to get excited by it. But if you use 480, 490, most of those photons will be absorbed by the FITSI molecule. So you look at this spectrum and you say, okay, this is how I choose my excitation wavelength. I have a light, I have an arc lamp, I will have a laser, which is illuminating in this wavelength range. Now, what do I do with the emitted photons? What color comes out of this one? Well, because of the energy loss, they will be towards the green side of the spectrum. You will not get blue photons out of blue excitation, you will get green photons out. But because of the many transitions, it's also spectrum. Most likely, you will get like 520 nanometer photons, but you will also get 510, 530, 540, 550, all the way to 600. So you will know that when you detect your fluorescence photons from FITSI, you will get signal from this approximately 100 nanometer range. And that's crucial to know, because if you have more than one label, and that also emits in this range, you will get mixed signals. You will be not sure anymore what molecule you are seeing. So you must know this spectrum. Now, the difference between the maximum wavelength of absorption and the maximum wavelength, the maximum probability wavelength of emission is called the Stokes shift. And the Stokes shift is the result of this internal relaxation of the energy structure of the molecule, which happens after absorption and before emission. And that's a blessing for us, because without the Stokes shift, we could not build a fluorescence microscope. Why not? Because I need an optical element that separates the excitation from the emission. Otherwise, the excitation light would go directly into the detector. So all I would see is a big, bright field of the light source. I need to separate. And the reason I can separate them is the Stokes shift. And the way it works is you illuminate your sample with blue light. The sample will generate green light, as we saw, and I have to separate them. And I separate them with this mirror, called a dichroic mirror, which has the properties of being reflected below a certain wavelength. So blue light will be reflected, and transmitted above that wavelength. So if I put a mirror here which has threshold and wavelength of, I don't know, 495 nanometers, it will reflect 480, which is the excitation light, but it will allow through 520 nanometers, which is the emitted form. So this way I can build a light path with these so-called dichroic cubes, where the excitation and emission are separated by this mirror. So I can have the excitation, the illumination path, and the detection path at 90 degrees. So I can build a fluorescence microscope. You can do this in upright mode or inverted mode. Of course, in each case, you will need these optical elements, where the main element, of course, is this dichroic mirror, which separates excitation and emission. They can also have these filters, which helps them separate a certain wavelength range for the excitation and a certain wavelength range for the emission. And that's what they look like. So inside that, there is an excitation filter, there is an emission filter, there is a dichroic mirror, and the way that works is that the excitation filter will select this wavelength range, the emission filter will select this wavelength range, and these are designed based on knowing these spectra, excitation and emission. How do we know these spectra? The manufacturers of these fluorescent dyes will provide them to you. Now, as I mentioned briefly, if you have more than one label, then you have to be aware of the fact that these are characterized by spectra, not individual wavelengths. So these spectra can overlap. If they overlap and you have a dye which is green, and then you have an orange one, and the yellow one, and the red one, you can end up with a big mess where various labels and various structures overlap with each other because they have overlapping spectra. Of course, you can make this situation a little bit better by designing proper filters for your dichroic cubes. You can make the band, the range of excitation and emission that these filters led to narrower. And if they are narrower, then you might be able to separate, or at least separate them better. But eventually, you are going to run into trouble. For example, in this case, CFP, cyan, and yellow fluorescence protein, the spectra really overlap a lot. And sometimes biologists will know much more about this than we do. You are limited of what color you can use. Your secondary antibodies come in certain colors, so you have to deal with the crosstalk. And then you will see an example from Ruben later, and I will also talk about it in Wednesday in advanced JIPipe. Optically, you have a way to handle this. I will tell you in a minute, but as an image analyst, you can also handle this by having control samples where you know how much overlap there is between the spectra in your particular sample, and simply compensating the measured images based on this knowledge. You will say, if I have a green image and I have a yellow image, I know that 13.5% of the green will leak into the yellow, and I simply take the green image, multiply it by 0.13 and subtract it from the yellow. So you can compensate for this if you know the optical behavior of the labels. So for this purpose, you have to have these control samples where you have one label, but you measure all the detectors. So you know how much of that signal appears in the erroneous channel. Another way to do this is that you image one color at a time. Does it slow you down? Of course, as many times as the number of colors. But as a trade-off, you will get clearer images because you measure them separately. It will not help if your label 2 also responds to the illumination by laser 1, because then even in sequential mode you will have signal in the other channel, and this is when the image analysis-based compensation will come in, which we will, like I said, talk about the latest on Wednesday. And as a closing, let's talk about a few most frequently used microscopy modalities. Of course, the cheapest and most widely spread is simply what we talked about so far, the wide field microscope, where you illuminate the sample with either transmitted light sources or fluorescence light sources, and you collect the light from the entire sample. You saw the example of the fungal hyphae, where I showed you the effect of the deconvolution. These images, when you take an image at any focal plane, will contain a lot of blurred parts because you detect signal from the part of the sample which is not in focus. And this means that part of the image will be sharp and part of the image will be blurred. What you're going to do with it? Well, one possibility is what I showed you, and a lot of manufacturers actually make wide field microscopes based on this principle, is deconvolution. You measure or you calculate the point spread function. You live with the fact that if you take a series of images along the optical axis, so-called Z-stack, each of these images will have a lot of blurred components because of the outer focus type, but then I'll run my deconvolution on it, and I have a nice, relatively sharp Z-stack of an image. This works perfectly well. It's a cheap solution. There is nothing wrong with it. A lot of biology labs here use the same method, like the image I showed you, which we are working on at the moment in a concrete project. But would it be possible, especially for very dense and scattered tissues, to have images just from a thin layer of the sample? Let's say you have brain, kidney, liver, whatever, scattered tissues. If I illuminate the whole tissue, first of all, I will not see through it. There will be so much scattering. Second of all, the out of focus light will be so strong that even the convolution may not happen. Well, one solution is that you just take a sharp knife and you cut your sample into thin slices. and put it in the microscope. And we use that, with the microtome, that you take micrometer size thick slices and then you image those on a regular microscope and then you put the images together. This has a problem that even though it gives you a very nice resolution along the axial direction, it destroys the sample. You cut it up, basically. So the question is, can we cut up a sample, a thick sample, without actually physically cutting it up? And the answer is yes, we can do this with the so-called confocal microscope. And the principle is extremely simple and exists since the 1950s. If I take an objective lens, illuminate a sample, and I detect the light coming out of the sample, for example, by fluorescence, it will be collected by the objective lens, and then go through a second lens and go through a detector. That's how wide field microscopes work. I will get light from every part of the sample because the illumination goes through the entire sample. The exception is two-photon microscopy, but that's a different story. I will get light because of fluorescence, for example, from every part. So the detector will get light from every part of the image. And that's why it's so blurred. Now, what if I'm clever and I say, I put a piece of metal in the light path of the emitted light, and I punch a small hole, the so-called pinhole. What happens then? What happens is that a lot of these light beams coming from the sample will hit the wall, and only those of them will go through this hole, which are, if I align this position properly, in focus. So I physically exclude the light beams, emitted light beams, from reaching the detector by putting a small hole in the light path, aligning it with the focal point of the lens. So only those light beams will go through it, which are coming from the focal point. What did I do? I excluded the out of focus light. The system is then called confocal because the focal point of the lens and the position of the pinhole have to agree. If the pinhole is out of focus, then it will let light through that is not out of the focal plane, so your whole confocal image will be blurred. But this is done by technician, and this is not something you usually have to deal with. So what do we have here? We have the objective lens. We have our thick sample. We focus along this brown line here. And we get, as an illustration, 3 different light beams with 3 different colors. And if I position the pinhole properly according to the focal point, only this orange, whatever color this is, will go through the hole. The green and the magenta one will hit the wall. Where this one came from, this came from the focal point. So I slice up my sample optically. Hence the name optical slicing. So confocal microscope does optical slicing. It is not a super resolution system, it does not increase your resolution. It will slice your sample axial. So you will get a better detailed view along the axial direction, not in the lateral. So, that's what such a system looks like, a modern system. But it has a problem. The problem is that there is a hole in the light path, which is the heart of the confocal system, but that's also the problem. Namely, when I illuminate the sample, with a laser, only the signal from the exact position of the illumination light spot will reach the detector. So I will see one, let's say 1 micrometer by 1 micrometer spot of my sample. I don't see my whole sample. I only see where the light is shining. So what I have to do is, I have to move my light beam across the sample to get an image of the entire sample. I have to scan the sample. So what I do is, I set up this illumination path as you saw here, but I will actually move my illumination light beam across the field, both in X and in Y direction. I go in X direction from left to right, turn the light beam around, go back, scan again, scan again, scan again, scan again. So this type of confocal microscopy does not give me an analog image that I can look at. I have to process the images. I have to collect these 1 micron by 1 micron patches into one image with a computer. Otherwise, I won't see, I will see one spot. Yeah? So this means that such as a confocal microscope, I have the illumination source, I have the detector, but I also have to have the so-called scanner. Now, if I take my microscope and I position this pinhole and then I scan my sample in the XY direction, I get an image of the sample in my focal plane. But because of the optical slicing, I don't see the rest of the sample. So what I have to do is also, I have to move the focus itself across the sample to see the entire 3 dimensions of the sample. So I scan in the XY, but I also scan in the Z direction, in the X direction, in order to get an image. So I go through the entire sample, XY, and then Z. So you need 3 scanners. You need an X scanner, which moves the light beam from left to right relatively quickly. The quicker the better, because the sooner you get through the sample. Then you need a second scanner, which moves then this light beam 1 line lower and scans again, and scans again, and scans again. This is the Y scanner. This is much slower because it waits until the light beam goes from left to right. And finally, I have to have a focus motor that will move the focal plane across the sample programmably. Now, of course, if you set the pinhole to a certain size, then you will get a certain level of thickness from which you collect the light. Now, typically, the size of the pinhole, first of all, determines how thin this optical slice is. The smaller the pinhole, the thinner the layer from which you get light. But you also get less light. And secondly, it's usually not measured in micrometers, because the size of the pinhole, the actual size in microns doesn't matter. What matters is how big is the pinhole relative to the airy pattern that we talked about. You remember that below resolution spots are transformed into an airy pattern, which is a centrally symmetrical interference pattern, and the optimal size, or the practically optimal size of a pinhole, is to cover the first maximum of the airy pattern. This will keep the microscope within the optical resolution range, and the first peak of the first maximum of the airy pattern, you can calculate this, read up on it, covers 88% of the intensity. So if you set your pinhole to that size, the size of the first airy maximum, you will only lose 12% of the light. and yet you are taking advantage to the maximum level of the optical resolution of the microscope. And this setting, when the pinhole covers the first area maximum, is called 1 area unit. And 1 area unit is now, to you, a very meaningful number. It means that you set the microscope, as I mentioned, to the practically optimal way. You don't care if it is 70 microns or 40 microns in the actual piece of metal. Actually, if I tell you the pinhole was 70 microns, you will have no idea how big that pinhole is relative to the area pattern, because I would have to tell you the magnification of the objective lens, the magnification of the condenser lens, the magnification of the lens in front of the camera, and so on and so forth. What you care about is this relative unit. Now, if you open your pinhole to 3, 4, 5, 10 area units, what you will end up with is simply a wide field microscope. So you can use a confocal microscope as wide field, but not the other. What is the disadvantage of closing the pinhole? You lose more and more light. So if I plot the resolution on the vertical axis, full width half max, and I plot the pinhole size in area units on the horizontal axis, then area unit 1 is where I would typically set my confocal microscope to start with. If I don't have enough photons, I start to open it. But as I open the pinhole and I get more light, I get less and less resolution, worse and worse resolution. This is a measured curve. And this, of course, also shows you that going below, I would say, 0.6 or so, or 0.5 area units doesn't make much sense. Because you do not get better resolution, but you lose light. And if you close your pinhole to 0, you get the maximum axial resolution, but you get no photons. So the theoretical optimal pinhole size is 0. But technically that doesn't work, so we usually start around 1. Opening it beyond 2 will turn you basically, like I said, into a wide field microscope. Sometimes that's fine. Sometimes you have a structure which is very thin, and you don't care about this Z resolution, but actually you want to see, let's say you want to see the entire nucleus. Then you open your pinhole, because the nucleus is big anyway, so you don't need the resolution. Now, axially, this is the curve. Laterally, you also get some improvement in the lateral XY resolution if you go below 1, but not much. And typically, when somebody says, yeah, confocal microscopes give me a better lateral resolution, it doesn't really. It gives you maybe a 20% gain of lateral resolution, but the price is very heavy. You lose a lot of photons. Your image gets noisy, and the analysis becomes a major pain in the neck. The effect of the area units is just shown here. As you see, we see more details with the half area unit than with 1. But this one is still analyzable, and it's always up to you and the microscopist and the analyst to decide what is the necessary Z resolution that you need. In terms of the axial one, if you open the pinhole, of course you lose confocality. So you now end up with out of focus blur in your images. And this is just a few comparisons of wide field on the left and confocal on the right. To show you the increased details because of the axial slicing, not because of the lateral resolution improvement. Now, I told you this is not a new idea. This came out, discovered by Minsky in the 50s when he was at MIT and was thinking about the same problem that I mentioned. How do I slice a sample without physically slicing it and destroying it? And he came up with the pinhole idea. So in his original drawings, you already have the pinholes. So there's nothing new about it. But the actual first commercially confocal microscope did not come out until 1994, so 40 years later. And the only reason we even know about this is that his brother-in-law, I think, was, or son-in-law, was a lawyer, and he said, yeah, you should patent this. Because what Minsky did was he didn't care, he worked at the MIT, and he built one, it worked, he showed it to his colleagues were like, fine, what do we use it for? And then they put it on the corridor, just as a piece of equipment. And then, but because of the patent, we know that it was him who discovered the whole structure. He never made it into a commercial product. And of course, the big problem was scanning. The optics was not there to scan the beam properly, but rather he moved the table on which the sample sat. And it worked, but it's very noisy because any movement will then misalign the sample. But at the end, it did work. But so that was then, and then this is what the system looks like now, where we actually scan the beam. And now a bit more about the inner principles of confocal microscopy. So you have a light source, but in addition to that, you have to have scanners. You have to have a scanner for the X and a scanner for the Y direction. As I mentioned, the X scanner has to be much faster than the Y because we want it to get through the width of the sample as quickly as possible. Now, because the light always goes through a point, it goes through the pinhole, the detector typically cannot be a camera. The camera would assume that you illuminate every pixel in the camera chip at the same time. This is a scanning system. So you would either have a camera but leave the shutter open the whole time until the microscope scans through the entire sample and then collects a lot of noise. Or you use a detector which is not a camera but a so-called photomultiplier. The photomultiplier tube, it detects photons from a very small spot focused by a lens that sits in front of it. I'll show you an example in a minute. Besides that, it has to have the fluorescent cubes, just like we already know from white field microscopy. There's absolutely nothing new here, except that the filters have to be strong enough to tolerate laser excitation if you're using lasers. And the scanning goes, as I said, goes X and then turns around, and either you collect photons on the way back or you wait until the laser beam comes back to the left side and then detect only left to right. This is called one-directional and two-directional scanning. Obviously, the two-directional scanning or bi-directional scanning is almost twice as fast because you don't have to wait for the beam to come back to the left. And otherwise, the scanning is determined by a clock which tells the scanner when to move from left to right and up down for the Y. But otherwise, what I really want to emphasize on this slide is that the beams come. Welcome. Fine spot. You will arrange your time later. What I wanted to emphasize here is that oftentimes is not realized by microscope users that when we say and when you receive images in the afternoon from Lumen, that we have image pixels. You have images made of little, usually square-shaped elements. A laser scanning confocal microscope does not stop at each point and collects photos. It moves continuously. You see these scanners, they move all the time. So this means that when we say a pixel is, 100 nanometers, it doesn't mean that the laser beam stopped at the 100 nanometer spot and we collected the photons, the fluorescence photons by the photomultiplier tube and then moved to the next pixel. It moves continuously. So what I get from the microscope is basically, what I get from the microscope is basically a continuous flow of photons coming out of the sample and converted into a current by the photomultiplier. But it's a continuous signal. So what is a pixel in a laser scanning confocal microscope? The pixel is a digital creation that we create. We take the photons that come in and we decide for how long we integrate them. We say the signal is coming in, it's a noisy signal, sometimes brighter, sometimes darker, and we say, please integrate this signal for a distance that corresponds to 100 nanometers. And I call that a pixel. But from the same signal, I can say, collect this for 500 nanometers. And now I increase my pixel size by a factor of 5 from the same signal by integrating this analog current, or voltage if you wish, for that much longer. But the signal is continuous. So I have a full freedom to determine how large a pixel is. The microscope will tell you a suggestion. So, okay, divide the image into 512 by 512 pixels. Or it will tell you the optimal pixel size based on the Nyquist criterion that we talked about much earlier is 145 nanometers. For that, set the pixel size to that. And you click on the button or you type in 145 and it's set. Because it will set the integration time to that. Okay? So I have full freedom. As we are scanning the sample, right? So how large of an area of the sample we scan is 100% of the velocity. I can say scan the whole sample. I can say scan 100 by 100 micrometers or scan 20 by 20 micrometers. Or keep the number of pixels the same. What did I do? I increase the resolution of the image, of the microscope, of the image. So what I can do with a point scanning confocal microscope is set the absolute optimal imaging conditions. No other microscope can do that. Because here I have full freedom to set anything up. Of course, practically, oftentimes, you will not be able to use the optimal settings because it will either take forever or it will destroy the sample because you need too much light and so on and so forth. But practically, you don't, but theoretically, you can set a confocal microscope to the absolute optimal conditions. And then this is the whole setup. Nothing new now, you know everything. And let's go then to the detectors. Like I said, since the light is going to a point, you cannot usually use a camera, but rather you use a photomultiplier. And this is what it looks like. So basically, a photon enters a vacuum tube and hits a metal electrode from which the photon energy will knock out one electron. Now, in front of this electrode called a diode, there's another one. And a negative electron will see a positive potential here, so it will accelerate towards the next cloud. And since there is a high potential here, several hundred volts, the electron will accelerate, and when it hits the second diode, it will have so much energy that it will knock out 2 or 3 electrons. These electrons will then see the third diode at a positive potential, so they will rush towards that. And these 2 electrons now will knock out 4, and then 8, and then 16, and then 32, and so on and so forth. What do? I magnify the signal. From one photon, I will get thousands of electrons. And then I put this across the resistor and then measure the voltage. So basically, I magnified, I multiplied my photons. That's the name photon multiplier 2. It does have a disadvantage that these diodes are not able to emit more than a certain number of electrons per event, so you can saturate them. If you have too much light coming into a photomultiplier tube, you will notice in your digital images that the microscope computer gives you areas which are completely white, completely saturated, have the same exact value, 255 or 64,000. That meant that you saturated your photovoltaic. You have to decrease either the illumination time or the laser power. Now, in terms of light sources, Minsky used an arc lamp, so you can use a candle if you want for confocal microscopy. For practical reasons, nowadays we use LEDs and lasers. Some systems still have arc lamps. The problem with arc lamps, for example, mercury or xenon, is that their emission spectrum is not continuous. So, for example, a mercury lamp is missing exactly at the wavelength range that you would need for FITC or Alexa 488, the most common wavelengths for excitation. Lasers have the advantage that their emission wavelength range is very narrow, so you can very precisely set them up to match the peak of your excitation spectrum. Disadvantage is that for each wavelength you need a laser separately. So a typical confocal microscope will come with 5, 6, 7 lasers. Nowadays the lasers are solid-state devices, so they are small, they are much cheaper, and they don't heat up so much than the old gas lasers used to. So they can fit into a box the size of a large shoebox. Of course, the laser lines have to be aligned, so they run along the same path to hit the same objective and the same spot. For this one, by the way, it's 100% crucial that we use optical fibers. The one we showed at the second or third slide today. Without optical fibers, these beams would run in the open and it would be almost impossible to align them. But this way, we align them into the entrance of an optical fiber, and then all the wavelengths just run down the same optical fiber which is connected to the microscope itself.
Sorry, I'm
Yeah.
just wondering,
Yes.
what's the difference between LED lamps and arc lamps? Is it only for the wavelength or?
The spectrum, yeah. So LEDs have these very nice Gaussian-looking spectra, and arc lamps have this ugly.
But mostly at LSM we have LEDs or?
Laser scanning confocals usually have lasers. Spinning disk confocals, which we will talk about in a minute, they have both. LEDs have less power and they have a spectrum, so you must have a filter. Lasers are so monochromatic that you don't need an excitation filter for the cube. It saves you a lot of light. So you only need an emission filter but no excitation. Sometimes they put in a cleaning filter just to remove some noise, but you don't have to. With LEDs, you must. So we have to. That's the LEDs are cheaper. Okay. Optical sectioning we talked about already. So basically, the whole collection of the data would be XY scanning and then moving the focal plane and then reconstructing the full image. And this is digital, this is computer-based, it's not really a live analog image from this technique. For the resolution, we have formulas. This is the real one. For a confocal microscope, you will see that if you compare it with the formula I showed you earlier, assuming that the two lenses are the same, it was 122 times lambda divided by NA objective plus NA condenser. If the two lenses are the same, which it is the case for fluorescence, this is the formula. And this is the formula for a confocal microscope. So you will see that there's only a 20% increase in the lateral resolution. So we don't use a confocal microscope to increase the lateral resolution, but rather to increase the axial resolution. This is the real formula. Don't remember it. I don't. Based on the area unit, where you can calculate the actual axial resolution. Any modern confocal microscope will give you this number as a suggestion of where you should set the Z step size, the distance between the Z layers, to be optimal. And one more word about the importance of not closing the pinhole too much. The reason is that no matter what your optical resolution is, no matter how good your system is, if your detector doesn't get a certain number of photons, you will not get an image that is reflective of the actual resolution. We need, as a rule of thumb, approximately 500 to 1,000 photons per object, for example, per molecule, before we can really reconstruct the image at a quality that matches the optical analog resolution of the microscope. Keep this in mind, that sometimes you need more photons, which you can achieve by making the detection time slower, so slowing down the scan, or increasing the laser power. It's always dependent on what the sample can actually tolerate. Of course, this also means that the pixel size in the XY direction depends on the numerical aperture, just like it did with any wide field microscope. So when it comes to lateral resolution, you can use the same formula. You will get a much better axial resolution with a confocal, but not as much of a difference in the lateral direction. Now, there is an improvement of the confocal technology called the AIR scan. Zeiss introduced this one, and we briefly talked about this with one of you in the break. Basically, the idea is the following. The size of the practical optimal pinhole size is 1 area. Yeah? Because that matches the first maximum of the area. But what if we don't put a pinhole there, but rather we put an array of detectors there? The middle of this would be corresponding to 0.2 units. And this way, I could detect the signal from all, in this case, 32 detectors. Actually, the last one, number 32, is not measured. Whatever. And then, when I integrate the signal from all of them, I would get the signal that corresponds to 1.25. What's the advantage? Well, up to this point, except that I have a very expensive detector instead of photometer. But the advantage is the following. If you remember the curve where I showed you the resolution, the axial resolution as a function of the area, and you remember it came down and then slowly flattened out below 0.5, there was not much change. What if I could measure the image at not just one area unit, not just one pixel's pinhole size, but many pixels, many pinhole sizes? What if I could measure the image at 1.3, 1.1, 0.9, 0.7 area? I would then be able to reconstruct this curve. And if I have this curve at a few points, I could extrapolate down to 0 pinhole size. Can I measure 0 pinhole size? Of course not, because then there's no photons, and you remember we need at least 500 to 1,000 photons to see the resolution. So what do? I put in an array of detectors, and then depending on how many of these integrate, I can get a signal from 0.2 all the way to 1.25 area. In a synchronous experiment, it's one measurement. They are all hooked up parallel. That's genius, yeah, because then I can calculate this curve, extrapolate it to 0 area unit, 0 the diameter, and I can get the optimal, theoretically optimal confocal image. And that's what you get. This is your confocal image. This is your area scan image. It's a square root 2 times increased resolution. And sometimes that's enough improvement in resolution to see something versus not seeing something. This unit costs you, I don't know, 50,000 euros or so, which is not much compared to the 1 million you paid for the microscope. It sits in a different box. It replaces the pinhole unit with this one, and you have an increased resolution, a really increased resolution microscope. Now, this is not an analog image. Okay, this is calculated afterwards. Takes a few minutes to do the. It's basically a deconvolution process. And these are just some comparisons: confocal versus Airy scan. And we go a little bit because we started a bit later, and we need like 10 more minutes. There's a big problem with laser scanning confocal microscopy. It's slow. It's slow because you scan the sample point by point, micron by micron. It takes up to a second, 2 seconds per image to collect your data. That's slow. If you are monitoring, for example, the images I showed you, those immune cells, macrophages, and neutrophils eating fungi, if I try to scan that with such a microscope, I will miss a lot of the movement. I will not be able to track the cells. The images I showed you were made with a spinning disk. What is the idea? The idea is that the reason laser scanning confocal is so slow is that we have to go through a pinhole. So we have to scan the image point by point. But what if I didn't have one pinhole? What if I had 1,000 pinholes? It would still be a confocal, but I would illuminate 1,000 points at once instead of one point. And that's what I do. I put an array of pinholes in the light path instead of one pinhole. Each pinhole is matched up with a lens which focuses the light on it, which is crucial for a confocal microscope. But of course, I cannot take as many pinholes as I need because then it's just a hole, big hole. So I need to make a pattern. Usually these are designed along a certain spiral pattern. And then I illuminate my sample with this, and then I rotate this disc. So now the illuminated points move a little bit. And then I rotate it again and again and again and again until I covered every point in the sample. And hence the name, because these discs actually spin. They spin 5,000, either 1,800 or 5,000 rotations per minute. And it takes, depending on the pattern, which is, like I said, a certain spiral, 2 to 3 rotations to cover the entire sample. So this means I have a microscope which is confocal, but it can scan one image, I would say 100 times faster than a point scanning. So if you have a live sample which moves a lot, and you need to track it, and you need to have a Z-stack, so to have the vertical events also detected, you need to switch to something faster, for example, spinning disk confocal. Of course, because we have not one but thousands of pinholes, I cannot use a photomultiplier. I need to use a camera. But the principle is the same. The camera shutter just has to stay open long enough to collect a signal from every illuminated point to cover the entire sample. But for a point scanning confocal, this would mean several seconds until the laser beam goes through. Here it takes like 30 milliseconds because you have so many pinholes. So you use a camera that has disadvantages. The sensitivity is not as good as for the photomultiplier. But you do get much, much faster image collection. Of course, if you don't set up your microscope properly and you do not spin the disk enough times to collect data from every part of the image, you will see this. And you see, okay, I have to leave my camera shutter open longer. So here I go from 1 millisecond to 5 milliseconds, 1800 rotations per minute, that's much better. And then I say 5 milliseconds because I need the sample fast, but I rotate my disk faster. Now, if I rotate my disk faster, my signal intensity goes down a bit, but I get a perfectly smooth image. And this is a practical advice. You will see this time to time. So then these are the settings that we deal with when we are planning confocal or in general microscopy. The collection or the selection of the lens, the formation of the microscope, pixel size, Z-step size, amplification, and further ways of increasing the signal quality. During the image analysis course, you will learn how to work on this better with confocal, with image analysis. But one thing as a message for this part of the lecture is that there is no such thing as an optimal image collection. Because if you keep in mind that you have to keep your sample alive, or at least you don't want to breach your label, you need to have a certain spatial resolution. You need to have a good enough signal to separate the signal from the background. And you sometimes need to collect the image fast because the cells are moving, for example. This phagocyte moves. These 4 conditions you cannot satisfy at the same time. There is what we call the pyramid of frustration, which shows you that if you are close to these, you are far from that. If you are close to these, you are far from that and that. You have to compromise. And then what you have to do is decide what is most important for that particular experiment. For example, if you need to keep your sample alive long enough because you are measuring live cell interaction, but the cells are moving and they are eating phagocytes, and then they are eating pathogens, then you need high speed and you need to protect the samples. Then you, within this pyramid, you have to be in the middle somewhere here, where you have a lot of specimen health, a lot of temporal resolution, but not so much contrast, not so much spatial resolution. You have to give up something. Now, image analysis, also artificial intelligence-based image analysis, helps you a lot. So you have to remember that you set up your conditions as optimal as possible, but you don't have to be perfect. You cannot be perfect, but you don't have to be either. And this we will talk about later. Now, just a few slides to give you a little insight into how we can improve microscopy further. The main goal, of course, is get images at high resolution, get images at fast, get images from large tissue or entire organs, and how do we choose our microscopy modality according to what we want to achieve. And the methods I will very briefly talk about are listed here, and they are different in terms of spatial resolution. You will see that all of them except one will not improve your lateral resolution beyond these 200 nanometers that we talked about. So you have to be aware of that any of these will not increase your resolution except for super resolution. But some of them will be very fast. For example, light chain microscopy, very, very fast. Some of them will be very slow, like point scanning confocal. Some will be faster, but much worse lateral axial resolution. So this table you can use to orient yourself a little bit on what you need to choose for your goals, assuming that you have availability of these techniques. And now in Jena we have a wonderful microscopy core facility as established by the Microverse Excellence Cluster, where you have almost all of these techniques. Some at the clinic, some at the city center and stuff. But just to give you a view, very quick view, for example, you can increase your axial resolution by taking advantage of total internal diffraction. If you think about it, when a light comes from an optically more dense environment into a less dense one, above the critical angle, it will refract completely. We learned about this before. However, in fact, in practice, this refraction is not 100% because a little bit of the light, approximately 100 to 200 nanometers, will penetrate into the second phase. And this is called the evanescent field. And this means that if I set up a microscope and I illuminate it at an angle above the critical level, but close to it, I will only illuminate my sample, which is on the other side of the copper slip, for about 100 nanometers. So without any confocal microscope, I sliced my sample optically to a 100 nanometer slice. But it's only one slice, the one close to the cover glass. But it's perfectly fine for those events where you want to measure, for example, the movement of motor proteins linked to the membrane attached to the cover glass. Or I can use, for the fluorescent excitation, instead of one photon, two photons. I can say I need one blue photon to excite this molecule, let's say FITSI or Alexa 488. What if I have two photons, both red? They would both carry half the energy that I need for the excitation, but I illuminate the sample at the same time with these two photons. The molecule would not care. It would absorb the two photons as it does one. But what is the advantage? The advantage is that longer wavelength photons scatter much less than shorter wavelengths. So if I have a green or blue illumination versus red, the red light will go much deeper into the tissue. So I can see through 100 or 200 micrometer grain size with a two-photon microscope, whereas a single photon scanning microscope would not see more than a few microns. Is it an expensive system? Yes, because such lasers, which produce these red photons, which arrive simultaneously at the sample, are very expensive. The laser itself costs you 150,000. Or, instead of illuminating at one pinhole's worth of light spot or 1,000 pinholes worth of light spots, I illuminate a whole sheet of light, a whole plane, called light sheet microscopy. I generate a light beam, which is not a line, but rather a two-dimensional sheet. And I focus that on my sample. What do I do here? I illuminate the entire cross-section of the sample. So if I have a camera perpendicular to this, I get an image of one Z layer at once. So this is even faster than spinning this confocal microscope. The Z resolution is not as wide because the Z resolution is determined by the thickness of the light sheet here. And that's limited, maybe to a micron. But it's fast. So if you want to measure, for example, the growth of a zebrafish, the zebrafish is a couple of millimeters, few millimeters, you will not see through it with a regular microscope. But the light sheet will shine through it, and since it's so fast, you can monitor the growth, or you can monitor the growth of plant roots at real time, because you are collecting the data so quickly. Also, since you are illuminating the whole sheet, but not the out-of-focus parts of the sample, the light damage is very low. So samples tolerate light sheet microscopy extremely well. If you look at the time stop here, this is almost 2 days long examination. We also have data from developing Drosophila larvae, which were imaged for 8 or 9 hours without any problem to bleaching or damage to the animal itself. Problem? Yes. the sample has to be transparent. Otherwise, the light sheet gets damaged, that gets destroyed as you try to shine through it. So instead of the original tissue, you have to clear them until they are transparent, and clear them without changing the biochemical structure of the tissue. But this has been a hot field for 10 years, and now there are protocols to clearing anything, from whole mouse brain to liver to kidney to everything. Now, as a last thought, our biggest concern was resolution. Yeah? And the resolution depended on the numerical aperture, and that determined the size of the points that you want. So if I wanted to increase the resolution of my microscopes beyond what is given until now, what I would have to do is change the size of my points that to something much smaller. But I cannot do that because diffraction limits everything to about 200 nanometers. So here comes the idea from Professor Hell and others, who they got the Nobel Prize like 10 years ago for this, stimulated emission depletion. And this means the following. Instead of using one laser spot, like we use for fluorescent molecules, I use two. I use the original, what I need for fluorescence, but I also generate a laser beam at a higher wavelength, which looks like a donut. What this laser beam does is that the molecules which are excited by the green laser, which is the normal fluorescence microscopy, those which fall within the orange excitation zone will be forced to be fluorescent at a much higher wavelength. So only those molecules which fall into this hole inside the donut will respond with the normal fluorescence photons. Those which are here will be going through so-called stimulated emission. That is, their photons will be in the red zone and they will directly go down without emitting the proper fluorescence photons. So this means that we only get signal from the inside of this donut. Now the beautiful thing is that the size of the hole here depends on the energy of the orange laser. The higher the energy, the smaller the hole. This of course means that to get very high resolution, you need a very high energy orange laser which will destroy the samples. There's a practical limit here to about 100 nanometers, down to maybe 70 actual resolution. But it is an improvement, yeah, compared to 200 nanometers. That's just another illustration. So this is the two lasers are all aligned, green, orange, and then this is the resulting spot, which is basically a much smaller point spectrum. And finally, the other possibility is that if you think about it, the whole problem was that two small objects, which are very close to each other, we cannot see because of the diffraction. And I cannot move the two objects apart because that will change the structure of the tissue. But what if I took these little molecules into only flashing light one at a time, or a few at a time? Then two molecules which are very close to each other, either one of them would emit light or the other one would emit light, but almost never both. Then I would see them. But I will see them on different images. In one image, one would be bright. In the other image, the other one would be bright. But then I would just take these images and put them together. And then I would have each and every molecule observed individually. The pattern could be fitted with a Gaussian function. And if I have at least 1,000 photons, like we learned earlier, I can fit a very good Gaussian here, and I will know exactly where the molecule is. The width of the Gaussian will be the resolution. If I can fit a Gaussian that's 20 nanometer wide, my resolution is 20 nanometers. What determines the Gaussian width? Or the quality of the fitting? The number of photons. So if I observe the sample long enough, like I collect 10,000 images, I will have my desired super resolution based on individual molecules. And that's it. That's what I want to tell you about microscopy.